thank you, Qian, thank you so much for introducing me and for inviting me to this meeting. And also, I like to thank all the organizers, as well as all of you in the audience for attending this talk so early, like on a Saturday morning in Asia. So as Qian mentioned, my lab is interested in using computational biology and also genomic approaches to study RNA processing and regulation. So we've developed new computational tools and genomic methods to study the variation and also regulation of alternative isoform expression in human and also mammalian transcriptomes. For today, I'm going to tell you about our recent work using large-scale RNA-Seq data to elucidate alternative isoform variation with a particular focus on the regulation and functions of pre-mRNA alternative splicing in cancer transcriptomes. So I'm required by my institution to disclose a significant financial interest and also outside consulting activities. So as we all know, human cells generate enormous amount of regulatory and functional complexities through a limited number of genes that are present in the genome. And post-transcriptional RNA processing and regulation is essential for establishing this regulatory complexity. So after transcription, the precursor messenger RNAs undergoes a variety of regulated processing and modifications before we can generate mature mRNA product that can be translated into proteins. And there are a variety of RNA level regulatory processes such as alternative splicing, alternative polydynylation, and also base modifications of RNA that can generate multiple mRNA isoforms from a single gene. And those different mRNA isoform products could have distinct regulatory properties in the cell, such as their localization, their stability, the rate at which they're translated into proteins. And also some of those mRNA isoforms could be translated into stable protein isoforms with divergent structure and functions. So this mechanism of post-transcriptional regulation generates a tremendous amount of functional and regulatory diversity in mammalian cells. And one of the really critical mechanisms for creating transcriptome and proteome diversity is this process called alternative splicing. So here I'm using a very simple cartoon diagram in which I have a 3 exon gene, and exon 2 is alternatively spliced. So after splicing, we can either include all 3 exons in the final mRNA product, or we can skip exon 2 and only have exon 1 connected to exon 3 in the final mRNA into 2 different protein isoforms with different functions. So in the last 2 decades, with different types of, with studies using different types of transcriptome technologies, we now have firmly established the prevalence and importance of alternative isoform variation in human transcriptomes. In fact, almost all multi-exon human genes produce multiple mRNA and protein isoforms through alternative splicing and also other types of alternative isoform variation. And this could include the simple patterns of alternative splicing where you basically have binary choices from 2 different versions of the spliced isoform, as well as their combinations that gives you those complex alternative splicing patterns. So this process is very important in regulating gene function in normal biological, in physiological conditions. And also aberrant alternative isoform variation is a major cause of many human diseases. And nowadays, RNA sequencing has become the most powerful and popular approach to study transcriptome variation, including alternative splicing. And RNA sequencing now can come in 2 different flavors. So short-read RNA sequencing allows us to generate a massive number of reads on a given sample. So this allows us to obtain highly accurate quantitations of individual exons and splice sites. And we can also identify mutations using short-read RNA-Seq data. At the same time, long-read RNA sequencing allows us to read the full-length isoform products. So the combination of these 2 different technologies really provides a very useful tools for us to elucidate the complexity of isoform variation across distinct biological states. And this picture just shows you that if you do RNA sequencing at enough coverage, we can start to identify cell-specific transcript isoforms. So this is actually a data set we generated a number of years ago on a pair of human prostate cancer cell lines. So one is a epithelial cell line, and another is a mesenchymal cell line. So if you look at the signals of RNA-Seq reads mapped to this particular gene SLAK, you can see that most of the exons are highly expressed in both cell types. But you have one exon, which is exon 13, which is very high in epithelial cells and is completely absent from the mesenchymal cells. And in the bottom, you have another more complex situation in this gene called CD44, in which you have a group of 9 exons in the middle that are highly expressed in epithelial cells, and they're almost completely absent from mesenchymal cells. And because of the rapid adoption of RNA sequencing technology in biomedical research, nowadays we really have massive amount of RNA sequencing data for human samples in the public domain. So for example, I think currently there are hundreds of thousands of human samples with RNA-Seq data in public databases. And some of the large consortium projects like ENCODE, Epigenome Roadmap, TCGA, and also GTEx have generated RNA sequencing data in a uniform manner across many human samples. So for example, the GTEx project has RNA-Seq data on over approximately 50 different tissue sites across up to 1,000 individuals. And the TCGA dataset has generated RNA-Seq data and also associated clinical information for over 10,000 cancer patients. So those large data sets create a lot of interesting opportunities for us to explore the regulation and functions of alternative isoforms. But this also requires the development of robust computational methods for looking at this kind of data. So this, as you know, this is an area of active research in the community. And over the last few years, we have developed some of the computational tools that are widely used by researchers to study RNA splicing. So here I'm highlighting 2 tools from our group. The first one is this tool called rMATS, which is a tool we developed for detecting and quantifying alternative splicing from large-scale RNA-Seq data. And this has been widely downloaded and used in the community. And in the bottom, I'm showing you another tool that we published more recently, in which we combine the deep learning with empirical RNA-Seq data to improve the precision of RNA-Seq splicing analysis. So we recently have developed and released this improved version of rMATS called rMATS-turbo, which uses a very efficient data structure that allows us to perform alternative splicing analysis in very large scale, massive RNA-Seq data very easily, efficiently across thousands or even tens of thousands of samples. So this tool actually uses 2 different steps to process the RNA sequencing splicing information. In the prep step, we basically use a very efficient data structure to condense all the information about splicing and also exon usage in the original RNA-Seq data into this very compact splicing graph definition. And this process can be done in parallel. So if you have tens of thousands of samples, you can run this in a distributed manner on the cluster or in the cloud. And once you collect all those individual splicing graph files, we have a second post step that we can combine any arbitrary set of splicing graph files and detect and quantify alternative splicing events. So this tool really has allowed us to look at splicing in very large data sets and to ask interesting biological questions. And so for the rest of my talk today, I'm going to just show you how we can use this kind of large scale data coupled with computational tools to study the roles of splicing in cancer. And as we know, for 2 decades, we know that the splicing patterns of cancer cells differs from the splicing patterns of normal cells. And this is not just the consequence of disrupted gene regulation in cancer. In fact, if you look at this picture that shows 8 different hallmarks of cancer cells, in every cancer hallmark, there are well annotated, well characterized alternative splicing events that can functionally contribute to those cancer hallmarks. So for example, alternative splicing of the CD44 gene, which I showed you a few slides ago, can activate the invasion and the metastasis of cancer cells. Alternative splicing of the CD45 molecule can allow cancer cells to avoid immune destruction. And also alternative splicing of PKM can allow cancer cells to have a dysregulated energetics. So there is because of the importance of a functional importance of alternative splicing in cancer, there is great interest of using genomic technologies and large scale data to elucidate how splicing is regulated in cancer, how dysregulation of splicing contribute to cancer cell phenotypes. And in the last decade, there actually has already been a number of studies doing large scale survey of splicing in cancer, primarily using data generated by TCGA project. So as we know, the TCGA project is a large international effort to characterize the molecular signatures and molecular profiles of cancer tissues. So the project has collected RNA sequencing data on over 11,000 cancer patients across 30 different cancer types, along with many other layers of molecular information, such as mutation, copy number variation, DNA methylation, protein expression, as well as clinical data. So this kind of resource has become a very useful, this kind of data has become a very useful resource for us to look into the complexity of splicing in cancer and their correlate with the clinical features and the genomic features. But one of the limitations of the TCGA dataset is that it's primarily on primary cancers. And the most lethal form of cancer, such as metastatic cancer, are not part of the TCGA study. So in this work, we're interested in studying how alternative splicing patterns change during the course of cancer progression. And we decided to use prostate cancer as our model to perform this analysis. And we developed a signaling pathway-driven approach to study how alternative splicing is associated with oncogene activation during the process of cancer progression. So this work is led by a very talented student who just defended his PhD thesis a few months ago, Dr. Yang Pan, in collaboration with a number of collaborators at UCLA, Dr. John Phillips, Dr. Owen Whitty, and also Dr. Douglas Black. So this picture shows you the natural history of prostate cancer, starting from normal cells in the normal, normal epithelial cells in the prostate, going to the primary adenocarcinoma, and also into those metastatic cancers. So during the course of treatment, aggressive prostate cancer can progress from localized and hormone responsive cancer to metastatic and castration-resistant prostate cancer. And in fact, about 20% of the tumors treated with the standard anti-androgen therapy would develop resistance associated with the emergence of small cell phenotypes with very poor prognosis. So in the past, if you look at those different stages of the prostate cancer progression, in the past, splicing in each disease state has been characterized, but there has not been any systematic study to characterize the variation of splicing across the entire disease spectrum. So we hypothesized that large-scale RNA-Seq data across the prostate cancer disease states provide an untapped resource for splicing analysis, allowing us to look into the regulation and function of splicing during prostate cancer progression. So to look into this question, we collected a large data set comprised of about 900 samples across different stages of prostate cancer progression. So this includes a data set from GTEx representing normal prostate, TCGA representing primary prostate cancer, and also tumor adjacent benign tissues. We also collected several smaller data sets from dbGaP representing from different sources, such as the Stand Up to Cancer, representing different forms of metastatic or aggressive prostate cancer, such as CRPC and also neuroendocrine prostate cancer. So we use this rMATS-turbo tool that I mentioned a moment ago to uniformly process this data set and generate discovery and annotations of splicing events. So in the subsequent study, we focused on exon skipping events, in which we have about 13,000 such events. And this represents the most well-characterized, the most well-understood type of alternate splicing event in the human transcriptome. And for each event, we used this metric called % spliced in to quantify the splicing pattern. So this is a proportion measurement. So it ranges from 0% to 100%. So if you have a complete exon inclusion, you get 0%, 100%. If you have complete exon skipping, you have 0%. And if the two isoforms are used at equal amount, then you get 50%. So the first thing we noticed is, we wanted to know is that whether we can use the alternative splicing metric to cluster those, data sets, samples from disparate, sources. And in fact, if we do this kind of clustering analysis, PCA analysis using count-based transcript expression levels, you get this PCA plot, which is really messy. So you can see that we don't have a very good separation between different disease stages. And this is somewhat expected because we know that these data sets are coming from different sources, different studies. So the batch effect would normally make the clustering very messy. But on the other hand, if you just look at the splicing-based measurement, if we use this psi value, which is a ratio-based measurements of alternative splicing, you can see that we just take the raw psi value and we can actually generate a very good PCA plot. So you can see 3 major clusters representing normal prostate, primary adenocarcinoma, and also the metastatic cancers. And you can see that different groups of metastatic samples coming from different studies are actually clustered together. So this actually is consistent with other recent work indicating that because when we measure splicing, we essentially measure the ratio of 2 different isoforms within a single gene. So in other words, the splicing measurement sort of have this very nice internal control. And there's a number of studies showing that this kind of alternative splicing metric provides a more robust molecular signature against the common issues such as batch effects in large scale RNA-Seq data sets. So another information that you can take from this study is that you can see that from the PCA clustering, there's obviously a significant difference between primary prostate cancer and also the metastatic prostate cancer. So the question is that what is the molecular mechanism that drives those changes? And to look into the molecular mechanism driving the changes of alternative splicing during prostate cancer progression, we developed this new approach called PEGASAS, which performs a pathway-guided analysis to identify splicing events driven by oncogenic signaling. So on this slide, I'm showing you the general process of the PEGASAS analysis. So assuming that you have a group of samples, in this case 900 samples representing patient tissues across different disease stages. So the PEGASAS approach performs 2 analyses in parallel. In one analysis, we perform alternative splicing quantitation using rMATS-turbo for every alternative exon in every individual patient. So you can obtain a data matrix for that. And in another analysis, we use the signature genes of important oncogenic pathways to infer the activity of the pathway. So the rationale for this analysis is that for most of those major oncogenic pathways, for example, the Myc signaling pathway, the pathway activation will change the expression level of a large number of downstream target genes. So if we look at the expression levels of the target genes in the patient tissue sample, we can use that to infer the pathway activity. So we can perform this kind of analysis for all the cancer hallmark pathways, and then we can obtain another data matrix, which is the activity of every pathway in every patient sample. So now we can have these 2 different data matrices. Then we can correlate pathway activity to alternative splicing level to identify pathway-related exon incorporation events. And we can also perform permutation tests to obtain p-value. So as an initial sanity check for this analysis, we apply this approach to 3 important prostate cancer-related pathways. So for the androgen receptor signaling pathway, you can see that in early stage of the disease progression, going into the primary cancer, you see increased pathway activity. But then when we go to the neuroendocrine prostate cancer, where the cancer does not depend on androgen signaling pathway anymore to grow, you can see a dramatic reduction in the pathway activity score. And this matches with disease biology. And for 2 other pathways, mTOR and Myc, you can see a gradual increase in pathway activity during the disease progression. And because the Myc signaling pathway is really critical for many human cancers, especially metastatic prostate cancer, we decided to do some additional analysis and validation of this Myc signature score using primary prostate cancer samples from the TCGA. So you can see that if you take all the prostate cancer samples from TCGA and also tumor adjacent normal samples, we can measure the Myc pathway score in different groups. So for the patients with either genomic amplification of Myc or the overexpression of the Myc gene mRNA level, you can see that their pathway scores are significantly higher than those from the benign, tumor adjacent benign tissues, which is expected. But interestingly, if you look at the primary cancers without Myc amplification and also without the overexpression of Myc, you can still see a significant elevation of pathway score compared to the benign tissues. And in fact, if we take the top 25% samples with high Myc signature scores, we can generate a survival plot which can separate these two groups of patients very well, with the patients having higher Myc signature scores, having poor prognosis. You can see this KM plot is comparable to the KM plot we generated based on the status of Myc amplification, and outperform the plot based on the status of Myc RNA overexpression. But if you look at the numbers, you can see that this Myc signature score allows us to capture a much larger number of patients compared to the status of Myc amplification and Myc overexpression. And this is consistent with several other recent studies showing that you can actually have hyperactivity of the Myc signaling pathway without amplification or overexpression of the Myc gene itself. So this again shows the importance of using this kind of and the utilities of using this kind of signature score to capture the activities of oncogenic pathways. So now we have established the validity of these pathway scores. We can look into our correlation analysis and we can identify alternative splicing events that are correlated with a particular oncogenic pathway. So on this picture, I'm showing you two alternative splicing events in two genes, SRSF3 and also HRAS, that are highly associated with the Myc signature score. So on the left side, I'm showing you the SRSF3. So x-axis is the Myc hallmark score. Y-axis is the psi value of this particular exon in the SRSF3 gene. And each dot is basically a particular patient. So you can see a very strong negative correlation between Myc score and also SRSF3 exon splicing. And you can recapitulate this pattern by just looking at the sashimi plots of this exon across the disease progression. You can see that the exon splicing goes down gradually, from primary tumor to metastatic cancer. On the right side, you see, this is another gene, HRAS, which is a oncon gene. And again, you see negative correlation between Myc score and also the exon splicing. Again, you can see a gradual reduction in the exon splicing levels as the disease progresses. So now with this kind of approach, we can really associate all the exons in the alternative exons in the transcriptome to a comprehensive list of 50 cancer hallmark pathways. So we can use this to systematically identify exon correlates of oncogenic signaling in prostate cancer. So this heat map shows the results of this analysis. So each row is a particular oncogenic pathway, each column is a particular exon, and the red and blue color shows exons with either strong positive correlation or strong negative correlation. So you can see that some of the pathways such as E2F, mTOR, and Myc have many correlated exons. And another thing which is interesting is that if you take those exons and ask, okay, what are the top gene ontology term enriched in their corresponding genes, you can see that for many of those pathways, the top enriched terms are, carry the terms such as mRNA binding or double-stranded RNA binding. So this indicates that many of those oncogenic pathways actually have affect alternative splicing of genes encoding RNA binding proteins. So we can actually look at this kind of heat map with a different display using this plot called a hive plot, which is a very nice illustration of those molecular interaction networks. So in this case, I have 3 dimensions in my hive plot. The first dimension are the 2,300 alternative exons that have correlations with oncogenic pathways. The second axis is a list of 7 selected pathways that are important for prostate cancer. And the third axis is a list of top enriched Go terms. So every time you have an exon correlated with a pathway, you draw a line. And if you look at this hive plot, you can see that Myc, E2F, and mTOR correlated exons, there are many such exons correlated with 3 pathways. And then if you look at what are the top enriched Go terms, they go to this term called the spisosome. So those correlated exons are often found in regulatory and core components of RNA splicing. So we thought this is really interesting, and we decided to take a closer look at the Myc signaling pathway just because of its ubiquitous importance in human cancers. But one thing that I want to emphasize here is that up until this point, we're just using the largest scale patient data to identify the correlation between oncogene signature with splicing. And as we all know, when we look at this kind of data, correlation does not mean causation. So the fact that you have exons correlated with Myc does not necessarily mean those exons are regulated by Myc. So in order to look at, to test whether those correlation of Myc activity really means, regulation by Myc, we need to perform genetic perturbation experiments. So our collaborator, Owen Witty at UCLA has established a very nice engineered human prostate cell lines with a regulated Myc expression. So the big, and then as a model system to model the progression of prostate cancer. So the basic idea is that you collect prostate, you collect prostate basal epithelial cells from the tissue donors. And then you use lentiviral transduction to introduce 2 oncogenes, Myc and AKT. So the AKT gene is introduced as a constitutively active form, while the Myc is introduced as driven by doxycycline. So then in this system, if you induce double activation of Myc and AKT, we basically can trigger the cells to undergo oncogenic transformation into this very aggressive adenocarcinoma phenotype. And at the same time, and we can establish cell lines in this way, but at the same time, because the Myc expression is driven by doxycycline, so if you take away dox, the Myc expression level is going to go down. So this gives us a controlled system to actually look at how alterations of Myc, protein could affect downstream transcriptome events. So we established the cell lines from 3 different donors. And then you can see that if you, in this system, if you take away doxycycline, Myc expression goes down, there is going to be a reduction in cell growth and also an associated change in signaling pathways. So we generated the RNA-Seq data on this, 3 cell lines with Myc on or Myc off. And if you look at the total gene expression, you can see that Myc is the most downregulated gene when you take away doxycycline, which is understandable, but there are many other genes that undergo differential expression. And if you take those genes to do a Go analysis, again, you see enrichment of genes encoding RNA binding proteins. And then if you take this, 3 cell lines with Myc on and off, if we take the RNA-Seq data to calculate the Myc signature score, again, you can see that when the Myc is on, the signature score is much higher. So this again validates the utility and also the accuracy of this Myc signature score approach. So now we have this data set. So it's essentially 3 cell lines, each with 2 conditions, Myc on and Myc off. And we want to use RNA sequencing data to identify differential splicing events between those 2 conditions as the events that are regulated by Myc. So this is actually a more specialized study design to study RNA splicing, in which we have paired replicates. So in this case, we have 3 samples as replicates. In each replicate, the Myc on and Myc off status is paired. And we actually have recently developed a statistical model to solve a very similar problem, which is to study allele-specific alternative splicing in human populations. So in this setting, we actually treat 2 alleles of a given individual as paired, and multiple individuals carrying the specific heterozygous SNP as replicates across the population. So we developed this statistical model called PAIRADISE to detect differential splicing events from those paired study design. And this uses a hierarchical model in which we can simultaneously account for the the read count on each particular, replicate and particular individual, as well as the variability among biological replicates. And we use this additive model to, to basically model the difference between the matched pairs. So this work just came out in American Journal of Human Genetics in September. And we have also, with the help of Dr. Qian Hu at Roswell Park Cancer Center, we have also made a bioconductor package for this tool, which is publicly accessible. So with this tool, we can apply PAIRADISE back to this Myc data set, and we can identify about 2,000 exon skipping events that are significantly differentially used once you perturb the status of Myc. And this actually includes this exon from the SRSF3 gene, which I showed you a moment ago. So remember, in that particular PEGASAS analysis, exon inclusion is negatively correlated with the status of Myc pathway scores. And in this particular analysis, you can see that when Myc is on, when Myc is off, we see 20% exon inclusion, but when Myc is on, you see only 3% exon inclusion. So this regulation matches what we identified from the PEGASAS analysis applied to the prostate cancer metadata set. And again, if you look at this particular list of 2,000 exons, again, we see RNA binding protein as a top enriched term. So all those studies basically consistently show that Myc regulates many alternative exons in the genome, and many of Myc-regulated alternative exons are present in genes encoding master regulators of RNA processing and splicing. So now we have these two data sets. We have the Myc-correlated exons discovered from the tissue analysis. We also have the Myc-regulated exons discovered from the engineered cell lines. So we can ask, do we have a very strong correlation? So if you do a Venn diagram, you can see the correlation is very, very significant. And so we identified the, we confirmed the Myc regulation of this exon 3 in SRSF3 gene, which encodes a splicing factor. We also confirmed the Myc regulation of this alternative exon in this HRAS gene, which we also identified from the PEGASAS analysis. So this is a really functionally interesting event. HRAS encodes an oncogene. So when Myc is off, this alternative exon is used, and this actually changes the C-terminal side of this protein, which actually gives you isoform that has, pro-apoptotic activities. But once you, have elevated level of Myc, the exon splicing gets repressed, and this actually will switch the isoform to an exon skipping product, which is anti-apoptotic and pro-proliferation. So this is the process of how you actually increase the oncogenic signals during the process of prostate cancer progression through alternative splicing regulations of oncogenes. And the other question that we want to address is that we have performed this PEGASAS analysis in the context of prostate cancer. But we know that Myc activation is really important for many types of human cancers. So is this Myc correlation signature conserved across different types of epithelial cancers? To address this question, we applied the PEGASAS analysis to two additional cancer types, breast cancer and lung cancer. And in each cancer type, you can see that we can identify thousands of Myc-correlated exons. And if you draw a Venn diagram for a triple intersection, we have about 500 alternative exons in the middle that are Myc-correlated across all three cancer types. So this set actually gives us some really interesting molecular targets to think about their function and also to think about their therapeutic utilities. So in fact, we have found that many of those alternative exons would encode peptides that can be processed by the antigen presentation pathway and presented at the cell surface for recognition by T cells. So this actually means that many of those Myc-correlated exons could provide useful targets for cancer immunotherapy. And this is an area that we're actively pursuing as a part of a research project in the National Cancer Institute Cancer Moonshot Initiative. So I want to come back to this SRSF3 exon, and I want to tie all those different things together. So this exon is is in the gene, using the SRSF gene, which encodes a master regulator of RNA splicing. So this is the splicing factor that recognizes RNA element in the RNA and then regulate the splicing of many exons across the transcriptome. And this, and this particular gene actually has an alternative splicing event itself, which is exon 4. And in the normal condition, if you want to actually generate a functional RNA and protein product from this gene, you basically need to skip this exon. And once you have exon skipping, the protein isoform, the mRNA isoform gives you full-length mRNA isoform that can be translated into the protein. But then this gene actually has an auto-regulatory mechanism. So if the level of SRSF3 protein is too high, this protein actually can bind back to the exon of its pre-mRNA. And this is a positive regulator of splicing. So when the protein binds to this exon 4, this will induce exon inclusion. But the exon 4 actually contains an in-frame stop codon, which is premature, meaning that it's far away from the end of the normal translation stop size. So as we all know, in mammalian mRNAs, if you have a premature stop codon, this kind of transcript is considered as a poisonous transcript, which will be recognized by mRNA surveillance mechanism called non-sensitivity decay. And the NMD mechanism can recognize those transcripts, degrade it, and this would lead to decreased SRSF3 protein. So if you look at this regulation, you basically have a negative feedback mechanism that allows the cells to control proper levels of SRSF3 protein. And additionally, if you look at the genomic region of this exon, if you just look at how this exon aligns to corresponding regions across many other different vertebrates during the last 500 million years of vertebrate evolution, you see that this exon is located in this ultra-conserved region with very little mutation, nucleotide changes during recent evolution. And in fact, over almost like 15 years ago, Stephen Brenner from UC Berkeley actually published this very nice paper demonstrating that the unproductive splicing of genes encoding splicing factors are often associated with highly conserved and ultra-conserved DNA elements in the genome. And this provides an evolutionarily conserved, functionally important mechanism to properly control the levels of those master regulators. So if you piece all those things together, what we are saying is that this exon 4 splicing is a really important mechanism for maintaining proper level of SRSF3, which happens to have oncogenic activity. So, but in the process of prostate cancer progression, you have activation of Myc activity, and Myc allows the cells to basically bypass this exon 4 completely. And so essentially the cells is able to bypass this evolutionarily conserved mechanism for controlling the SRSF3 protein level. And this leads to a oncogenic and aggressive phenotype. And in fact, when we look at the cell lines with Myc turned on, you see much higher level of SRSF3 protein level, which is going to have a downstream functional impacts. So I want to summarize my talk here. So today I talked about the use of RNA sequencing to study alternative isoform variation, including alternative splicing. So we have developed rMATS-turbo, which is a computational tool for ultra-fast splicing quantitation across large-scale RNA-Seq datasets, and this is freely available from this GitHub repository. We have developed PEGASAS, which is a big data framework to profile alternative splicing landscape across cancer progression, and also identify splicing events driven by oncogenic signaling pathways. We show that splicing machinery is a major target of oncogene-driven alternative splicing prostate cancer, with a Myc-regulated exons enriched in regulatory and core components of pre-mRNA splicing. And then finally, I just want to say that this PEGASAS approach is really a generic approach that can be utilized to mine large-scale transcriptome data to connect the changes in pre-mRNA alternative splicing with the molecular alterations that are common to many cancer types or even non-cancer related diseases. So we're really interested in applying this approach to study the regulation of splicing as well as other types of alternative isoform variations in cancer transcriptomes. So I want to end my talk here, and I'd like to thank the people in the lab who contributed to this project. So the PEGASAS work is led by a very talented graduate student, Yang Pan, who just defended his project, and he has been the main driver for for a variety of our recent work on studying isoform variation in cancer transcriptomes. The development of the PEGASAS tool and also the PAIRADISE tools for looking at RNA-Seq data is a joint work from several other students and postdocs in the group. And I also like to thank our collaborators, especially Dr. Owen Whitty and also Dr. John Phillips, as well as Dr. Douglas Black for collaborations on the prostate cancer project. And also I want to thank Dr. Song Liu and Dr. Qian Hu from the Roswell Park Cancer Center for collaborating with us in developing the PAIRADISE bioconductor package, as well as a variety of other tools, projects that are ongoing. And I also like to thank various funding agencies for supporting our work, including the NCI Cancer Moonshot Program, the Park Institute for Cancer Immunotherapy, as well as the NCI Immuno-Oncology Translational Network. And we are currently performing, we are very interested in using those tools, including both short read and long read sequencing, and also bulk and single cell technologies to study the complexities of RNA processing and regulation in cancer and many other diseases. So if there are any people in the, if there are any postdocs or students in the audience interested in looking for, postdoc opportunities or more like, opportunities as a computational, biologist, feel free to, reach out to me, and I will be happy to talk about opportunities in the lab. And thank you. I'll be happy to take some questions.
Thank you so much, Dr. Xing. Very great talk and very useful information you have passed, and those very useful computational tools. We do have many questions in the chat board. I wonder if you can stop sharing and look at those questions.
Okay, all right. So, okay.
So the first question is from Louis Tian. Myc is a master regulator that have many downstream effectors. Do you think regulation on alternative splicing is direct or indirect?
Yeah, so this is a great question. So, by the way, I really want to thank the, well, I guess Louis Tian, because we have been using some of his tool, bioconductor packages and tools in our research, especially related to single cell analysis. So this is a great question. So Myc is a master regulator, so you can have many downstream impacts. So the question is the regulation of alternative splicing direct and indirect? I think at this point, my honest answer is that we don't know, because I think there's a variety of possibilities where Myc can regulate splicing events. So for example, we have shown that Myc can regulate the splicing or expression of genes encoding RNA binding proteins. And those RNA binding proteins could in turn regulate other downstream splicing events. So this will be an indirect effect. At the same time, as a master regulator, Myc could change the local chromatin states and epigenetic states. And we know that many splicing events occur in a co-transcriptional manner. So it's entirely possible that Myc can regulate some exons through a direct epigenetic mechanism. At this point, we don't know, but we are in the process of performing large scale, screens to look for, for example, RNA binding proteins that mediate or control the regulation of those Myc target exons. So I think we should be able to have some, answers into your question, hopefully in the very near future. So that's the first question. And the second question is, any thoughts on the general functional effect, of alternative splicing be regulated? Will they mostly generate unproductive, protein, so that alternative spliced RNA in the end reduce the protein expression? So I think if you look at the two examples I showed in my talk, I have this SRSF3 example, which is a Myc-regulated alternative splicing coupled with a poisonous cassette exon, coupled to non-sensitivity decay. So in this case, the Myc regulation essentially occurs, through splicing, but affecting the levels of RNA. But I have another example, HRAS, which is, a case where the splicing event basically changes the protein product, on the C terminus. So I would say that both, scenarios are present in our data. I mean, we do find that in the 147, events that are kind of Myc-correlated and also Myc-regulated, many of them actually encode poisonous cassette exons. So I would just say that the first mechanism is a fairly prevalent mechanism, but I think other mechanisms also apply, including the effect on the protein. So the second question is, can you comment on quality control of RNA-Seq experiment to ensure successful splicing study at such a large scale? Can data from FFPE samples be used? And what's the common read depth? So, so there's several ways to answer this question. First of all, when you look at the splicing, especially if you're interested in the patterns of final isoform product in the in the mRNA pool, then you want to be able to look at analyze a data set in which you have you want to analyze samples where you have good RNA quality. So I think RNA integrity is really important for this kind of analysis, which makes it a little bit harder to do good splicing analysis using FFPE samples in which the RNAs are so degraded. And but to look into the RNA, the issue of RNA quality control, you can look at a variety of QC measurements on the data. So for example, what's the coverage of the RNA sequencing reads across different genomic regions, intronic, exonic regions, and what's the coverage plots across, from the 5' end of the transcript to 3' end of the transcript. And if you have high quality data, you want to look at you want to be able to have a more or less a uniform coverage. And the last question, what is the common read depth requirement? I would say that it's, this is really dependent on what's your goal. I mean, if your goal is to primarily look at gene expression, but you want to look at the splicing as well, I would say that a typical coverage of 30 to 60 million short reads across on the sample is probably what most people are doing. But if you are primarily interested in RNA splicing, then you probably want to have much deeper coverage. And in fact, Dr. Chine from University of Michigan had a review article on, on the use of RNA sequencing for cancer transcriptome profiling. And he has this sentence in the paper, in the review article, saying that to do splicing analysis, you need to have maybe over 100 million reads on a given sample. But you can see that the TCGA dataset does not have that kind of coverage, but we can still be able to find a lot of interesting events.
Okay, we have a question from Xue. Can rMATS-turbo be applied to long read data? Will longer read length affect the calculation of ratio? So this is really interesting because, I would say that there's a short answer, which is no. rMATS-turbo is developed for short read data, and it does not apply to long read data. But I would say that if you look at the overall, kind of history of transcriptomics in the last 20 years, we started from long read sequencing, which is cDNA and EST sequencing. We went to microarrays, we went to short read sequencing, and now we're back to long read sequencing. And so I would say that there are lots of insights that we, developed, 20 years ago on, EST analysis, which could actually be applied to, long read analysis. But obviously, there are many useful tools developed today that specifically deal with the features and also technical issues of either PacBio and nanopore long read RNA sequencing. And I'm pretty sure that some of the speakers at this conference probably would have talked about long read analysis. This is an area that we are very interested in. So I would just say that stay tuned. We're very interested in developing approaches for long read analysis. Oh, one more question from Xizhen. How do meta-analysis of alternative splicing use RNA-Seq data from GEO database? Do you have the practical tutorial?
I mean, technically, rMATS-turbo can be used for doing that. So we have never done that ourselves. But the computational framework allows us to do that because we essentially just take each BAM file, convert to the splicing graph file, and you can do this on all the 100,000 samples. And you can actually look at splicing in this way. The one thing I would say that, rMATS-turbo has this very efficient data structure, which makes it very fast. At the same time, it also has a benefit over some other approaches which are using alignment-free approaches to perform very fast splicing quantitation. Because when you do alignment-free analysis, you basically have no ability to detect novel splicing events and novel exons. But the rMATS splicing graph actually captures the entire information about splicing, including novel events in the data. So I think in principle, it could be used for doing that, but we have not done that. But on the other hand, I would say that there are other efforts in the community which also perform large scale quantitation of splicing using GEO level data set. For example, there's this project called Recount or Recount2 that does just that. So I think if you're interested in just grabbing read counts and also look at splicing using very massive data sets like GEO, I think there are also existing resources that you can use.
One question from Julie: how to deal with the batch effect in analyzing large data sets from different data sources?
Yeah, so I think there are two ways to answer this. One is that if you look at our prostate cancer data set, we do have data set from very disparate resources, but you can see that when we cluster the samples based on the splicing levels, we actually get a very nice sample clustering that matches their biological states. So I would say that the one information from that is that the splicing measurement seems to be a little bit more resistant towards batch effects in the large data set. With that said, it does not mean that you don't have influence by batch effects. And we recently published a study in American Journal of Human Genetics, in which our goal is to look at genetically regulated splicing in the human brain. So in that study, we looked at 1,200 human brain samples, and we basically used basically, for example, surrogate variable analysis and other approaches to control for confounding factors in the study. So I think we can borrow information, techniques that people developed for batch effects removal and also confounding factor analysis using gene expression data.
So since we still have time, there's
I think there's one last question, so maybe I just should answer that,
okay.
and then we'll be
last
done.
Question from Qian. The phenomenon that autoregulation of splicing factors was also seen in my data sets. Genes with differential spacing events were mainly proteins involved in alternative splicing.
Yeah, I guess this is a comment, not a question. I would say that, yes, I agree with you. So many splicing factors have alternative exons, and the alternative splicing of those splicing factors play an important role in regulating the activity. So in the case like SRSF3 and also some other splicing factors, the alternative splicing is correlated, is coupled to, is basically a poisonous cassette exon, which would subject the transcript isoform to degradation. But in some other cases, for example, the splicing factor RBFOX2 has two isoforms that are both protein coding, but then they have different cellular localization. So when you have one isoform, it's a cytoplasmic isoform that primarily binds to 3' UTR to regulate RNA stability. But once you have another isoform, it becomes a nuclear form that is involved in regulation of splicing. So I would just say that the one thing that which is really important, which is still a largely unsolved questions, is that what's the functions of those isoforms? I mean, if you look at the history of transcriptomics over the past 20 years, finding novel isoforms has never been a challenge. We always find many isoforms. And nowadays with long-read sequencing, we're going back to this old days of, we're finding so many isoforms. That's not, I feel like that's never a challenge. The challenge is that what are their functions and how are they regulated? I think the question of function is something that is really important, and we need to think about the systematic ways of probing isoform function.
Great, thank you so much, Dr. Xing. Like you feel so smart, like you talk fast and you explain everything so clearly. And your topic is really like triggering great interest for the attendees. Thank you so much for giving this keynote talk today. And I hope you have a good night.
Yeah, thank you so much. It's really my pleasure. Yeah, thank you all.